Hey guys, I'm Abhijit and today I'm presenting the 7th episode of Basics of Medical Science, and in this episode I'll be presenting the nerve tissue. Now nerve tissue refers to a group of organized cells those controls the body's movement along with the muscles, and they receive the signal from the different parts of our body like touch, pressure, temperature and pain, and those signals are sent to our brain, and from our brain the second signal which was being sent to the muscle tissue for accordingly movement or for muscle contraction. Nerve tissue has a role in controlling the vital body function such as digestion, pumping of heart and respiration, etc. There are two types of nerve tissue, neurons and glial cells. Neurons carry the signal as electrical impulses, and whereas glial cells have the function of supporting and protecting the neurons. Moreover, it acts as an glue to the neuron. Now let us look at the structure of the nerve tissue, that is the anatomy of the nerve or the neuron. Neuron consists of dendrite, cell body, myelin sheath and axon. Now dendrites are the highly branched structure as you can see here in this picture, and that is responsible for receiving and processing the information from the other neuron or muscles. They are connected to the cell body. Now cell body of a neuron consists of the nucleus, cytoplasm and cell organelles like any other human cells. Axon are the long projections emerging out of the cell body, and the endpoint of the axon are known as axon terminal. These axon terminals are responsible for communicating with the other neurons as well as muscles. Myelin sheaths are the protective fatty coating surrounding the axon, similar to the insulation around an electrical wire. Neurons receive signal and those are transmitted across the axon and at axon terminal converted to the chemical messengers. These chemical messengers are called neurotransmitters. They bind to the receptor present at the next neuron or at the muscles. Neurons respond to the stimuli like touch, pressure, temperature and pain, and this stimuli gets converted to the electrical signal which transmit across the neuron, and finally it gets converted to the chemical signal at the axon terminal. So now the action potential is the basis of this electrical signal. So let us understand the details of the action potential. The initiation and the transmission of the nerve impulse is called action potential. Each action potential results from the movement of ion across the nerve cells, and all this ion enters into the nerve through a small pores which are there, and these are called the ion channel. You can see the cross section of the nerve, and in this there are ion channels which are present, separate for calcium ion and the sodium ion, and these ion channels are stimulated by the receptors. If the receptors are stimulated, this ion channel open up and allow the passage of the ion from outside environment to the inside the cell. Another important structure in the cell membrane is the ion pump. This ion pump pumps out the ion from inside the cell to the outside environment. During the resting state of a neuron, the inside environment is negative, that is minus 70 millivolt, and external environment is positive, and that is how it is called polarized. And the bicarbonates ion are the negatively charged ion, which are along with the protein molecules. They remain inside the cell. When the cell is unstimulated or at resting state, there is an accumulation of the positively charged ion or cations outside the cell membrane and the negatively charged ion inside the cell. Now sodium and calcium channels on the cell membranes are closed at the resting state of the neuron. At the same time, the sodium and calcium pumps, they pump out sodium and calcium ion outside the neuron, and thereby the outer surface of the neuron are positively charged, and the internal environment they maintain a negative charge of minus 70 millivolt. Now, chloride ion is also negatively charged ion, and whose channels are always open on the cell membrane of a neuron. So these ions can easily enter from outside to the internal environment of a neuronal cells, and then they maintain the negative charge inside the neuronal cell minus 17 to minus 90 millivolt, and it is called the resting membrane potential. When the cell membrane is stimulated, the sodium channel opens up and allowing the accumulated sodium ion to rush inside the cell through the sodium ion channel, as you can see here in this picture. As a result of this positively charged sodium ion moves inside the cell, so internal environment converts from minus 70 to minus 30 millivolt. At this stage of depolarization, the calcium ion channel opens up. Calcium ion channels are the voltage gated, and that's why, because of the fluctuation of the voltage from minus 70 to minus 30, the calcium ion channel opens late. And this entry of sodium and calcium helps the rapid shifting of the internal environment from minus 30 to plus 30 millivolt. Now the neuron is completely depolarized. Now there is another event that can take place after depolarization, and that event is known as the repolarization. And here what happens, the calcium and sodium ion channels are then closed, preventing further entry of this calcium and sodium inside the cell, and at this stage the sodium and calcium pump they start, and once again they restore the internal voltage from plus 30 to minus 70 millivolt. And this process is called repolarization. In case any stimulus to the receptor gives inhibitory signal, in that case the chloride ion channel in the cells that opens up, and then there is an entry of the chloride ion from outside environment to the internal environment of the cell. That makes the internal environment of the cell more negative. So that is why it is called hyper, means excessive polarized state, and at this state the internal environment becomes minus 110 millivolt. Now let us understand how the signal propagates from one point of the neuron to the other point. The wave of the depolarization and repolarization moves from dendrite to the axon terminal, and at the axon terminal then the calcium channel they open up, and entry of the calcium ion takes place at the axon terminal. After this event, both the electrical signal and the calcium ion they help the pushing of the neurotransmitter at the synapse. Now let us understand the definition of the neurotransmitter and receptor. Neurotransmitters are the chemical, those are releases at the synapse and transmit impulse to the other neurons, muscles, organ or tissues, whereas a structure on the surface of the cell that selectively receives or binds a specific substance or neurotransmitters are called receptors. This is the diagram of the neurotransmitter, where neurotransmitter you can see is being released from the presynaptic nerve terminal to the and binds to the receptor in the postsynaptic nerve terminal. You can also see a diagram of the ion channel which are present on the cell membrane. Now there are some important neurotransmitters like acetylcholine, which is released between the neurons and the muscle cells. Acetylcholine helps in the contraction of the muscle. Moreover, acetylcholines are also released in the synapse of the brain. Second, amino acids are the second type of neurotransmitter are the amino acid neurotransmitter. For example, gamma aminobutyric acid, which are also called GABA. These GABA are present at high concentration throughout the nervous system and is a major inhibitory neurotransmitter. Glycine is also another major inhibitory neurotransmitter, and these are present at the spinal cord. Glutamate is an excitatory neurotransmitter. These are found throughout the nervous system. Apart from that, there are many neurotransmitters which are known as biogenic amines like norepinephrine, epinephrine, serotonin, dopamine, and histamines. When this neurotransmitter gets released at the nerve ending or at the synapse, immediately they need to pass the synapse and bind to the receptor. But at the same time, the synapse needs to be empty very fast. Otherwise, this blocks the other signal which may immediately come across. So what are the events that takes place in the synapse? The first event is the receptor binding of this neurotransmitter to the next neuron and thereby which propagates the signal. There are certain neurotransmitters which diffuses out of the synapse to the external environment out of the synapse, and third type of removal process of the neurotransmitter are the enzymatic breakdown. There are certain enzymes such as acetylcholine which breaks down acetylcholine esterase which break down the acetylcholine. There are also enzyme like catechol or methyltransferase which breaks down the neurotransmitter like norepinephrine. Then there is another process by which synapse gets empty, that is the reuptake mechanism. There are some transporters which are available at the synapse. They actively transport the neurotransmitter from the synapse back to the presynaptic neuron. And finally, there is autoreceptor binding. That means upon release from the presynaptic neuron, some of the neurotransmitter goes back to the presynaptic neuron and binds to the receptor over there, resulting in the break in the release of the neurotransmitter. So this also autoreceptor, they act as an brake, and that is why they are called autoreceptor. So this is all about the neuron and the nervous system. In the next episode, I shall be covering the classification of the nervous system, and if you have any question or suggestion, you can get in touch with me at abhijitdas61@gmail.com or my Instagram, Facebook or Twitter or Q account. Signing out until I meet you in the next episode. Goodbye.